Hi everyone, so in today's video we're just going to be studying deeper about the DNA. We'll look at the location, the structure, the functions, and also the brief history. So let's get to it. Okay, so let's study for the day. We are going to focus on just one nucleic acid, which is deoxyribonucleic acid, which means just the DNA. So in today's notes, we're just going to be studying the DNA information about all of it. All right, making my notes for myself. As I said, first, I just want to know generally what do I remember about DNA, even from the previous video. Okay, so we remember that DNA, or rather deoxyribonucleic acid, is made up of nucleotides, okay, which are the building blocks. So it is made up of nucleotides. And then we remember that it contains nitrogenous bases. Obviously, we remember also that it's got the phosphate, it's got the sugar, but this one, important. It contains nitrogenous bases. Okay, let me use my different colors now. It's adenine, that's the first one. The second one is thymine, which they pair together. We remember that one, and they should be the same amount since they pair together. It's cytosine, and the last one is guanine, and these two also pair together. Okay, so now just quickly reminding myself that DNA is made up of nucleotides, and those are the building blocks. It contains 4 nitrogenous bases, which is adenine, thymine, cytosine, and guanine. Okay, and then you also remember then it's got a phosphate group and a deoxyribose. Then the most important thing about DNA is that it carries the genetic code. It carries the genetic code for protein synthesis. So all the proteins that are going to be formed in your body. And as we continue, you'll see we learn more that with proteins, you've got the building blocks, amino acids, and they are responsible for building your entire body. So because of DNA, the amino acids then get, instruction, which is the genetic code. Right, so let's just go through this again. With DNA, deoxyribonucleic acid is made up of nucleotides. It contains nitrogenous bases adenine, thymine, cytosine, guanine. And most importantly, its function is that it carries the genetic code for protein synthesis. Okay. We go now to the brief history. Okay. It's very few things that you must remember about the brief history of DNA. They don't really ask much about it. Okay, so I'll be using colors to also just remind the scientists who were involved in the formation of DNA. So the first one, who is Rosalind Franklin. Rosalind Franklin, who worked with Maurice Wilkins. Okay, so Rosalind Franklin and Maurice Wilkins worked together when they were researching. They researched the structure of DNA in 1952. Okay, so with DNA, I need to know that Rosalind Franklin and Maurice Wilkins were the first guys who researched the structure of DNA, and that was in 1952. Then we go to the second guys who worked on it, who is James Watson and Francis Crick. These ones then did some research. Who did research? Okay, let me fix that. Who did research on DNA using Franklin's work and proposed a 3D double helix model for DNA in 1953. Right? Let me just finish it off and then we'll go through it again. Then now it's towards the end when now Watson and Crick actually now received a Nobel. Okay. Watson and Crick. Okay. Watson and Crick received a Nobel Prize in 1962. And unfortunately, by that time, Franklin had already passed away. And it's so sad that actually a Nobel Prize can only be received by 3 people. So I don't know where that put Wilkins actually. But now that Franklin had passed away, then definitely Wilkins had to be included because he also worked on it. So it was definitely going to be a battle if Franklin was still alive. Okay, let's go through it again. So with the brief history of DNA, we know that there was Rosalind Franklin, Maurice Wilkins. The two of them researched the structure of DNA in 1952. Okay, then James Watson and Francis Crick did some research on DNA using Franklin's work. Managed to come up with a 3D model of DNA, which we now refer to it as a double helix. And this was in 1953, so just a year after. And then when their work was then recognized and accepted as it is even today, Watson and Crick then received a Nobel Prize in 1962. And as I mentioned, Franklin had already passed on due to cancer. So this, if you can just get this information, you are fine. Just knowing who were the main scientists in which year, and then who actually now came up with the model, because they are important. They used the work just to get the model. If you can know that these two came up with the model in this year, then you're fine. And then if finally just knowing that they received their Nobel Prize in this year. Okay, now let's look at the shape of DNA, or rather the model that Watson came up with. Kudos to those guys, because now we've got something to actually materialize in our life sciences, because our DNA is very, very small. We can never see it, but because of the model they came up with, we are we're flourishing. Okay, so with the shape, I'm just going to do my quick notes also. The shape of DNA consists of, very important when I use my color, two strands. Okay, as you remember, even I think on our last video, we do have a picture, so you can go check it up, but I'm also going to bring you another one now. So the shape of DNA consists of two strands that are joined together. Okay, I don't know if you can remember, by weak, weak hydrogen bonds. And I did say they need to be weak so that during DNA replication on our next video, it can be able to unwind. Right. Okay, so the shape of DNA consists of two strands joined together by weak hydrogen bonds and twisted. It has been twisted spirally. All right, and then we have then our scientific word for that. Thanks to Watson again, we now call it a double helix. Okay, so it's a DNA double helix. So what I want you to get from this part is that your DNA, remember DD, it's got double strands. So it's two strands, and those strands have been joined by the weak hydrogen bonds so they can be able to unwind. And because it's in a twisted structure, we refer to it as a double helix. Let me put up the picture that I want to use for my notes, and this is exactly what I'm talking about, just this one. Because it is twisted, you can see it's going around. It's referred to as a double helix. Okay, it's all these nitrogenous bases have been joined by the weak hydrogen bonds so they can be able to unwind. And then when you look at it like this now, it has been untwisted, now it's unwinded, so it's just a double strand, just like that. Okay, so you need to be able to see the difference. And please, most importantly, this is the double helix. This is just a double strand. Okay, double helix because, go back to our notes, double helix because it's twisted spirally. All right, and then double strand when you can see both strands visibly. Okay, and then most importantly, weak hydrogen bonds so it can be able to unwind. All right. All right, let's go now to our last part, which is the location. Where do we find the DNA? All right. Now I've got two places, and I hope you remember your cell, which you learned in grade 10 and a little bit in grade 11 and somewhat in grade 12. I will try my best to go through that and just get a video for that. So we can just go through the cell so we can see the nucleus and everything, all the organelles inside. Okay, so since we are here focusing on DNA, we've got now two locations. Okay, we've got firstly DNA found in the nucleus. So this one is found exactly inside the nucleus. And because it's found inside the nucleus, we refer to it as nuclear DNA. Okay, the word helps us remember there. Okay, then we've got DNA found. Okay, sorry about that. DNA found outside the nucleus. Okay, DNA found outside the nucleus. We've got two of them. We've got one that is in the mitochondria, so we'll call that one mitochondrial DNA, which means if it's outside the nucleus, then it's in the mitochondria. And then the other one, which is found in the chloroplast, therefore it will be chloroplastic chloroplastic DNA. Right. And these are your two organelles that are found outside the nucleus. They are just there in your cytoplasm, your mitochondria, and your chloroplast. And then these then are referred to as extra nuclear DNA. Okay. So again, for your location, two places, pin the location. Okay. DNA found in the nucleus is referred to as nuclear DNA. And then DNA found outside of the nucleus, it can either be in the mitochondria, therefore it will be mitochondrial DNA, or in the chloroplast, and then it will be chloroplastic DNA. And then they are both referred to as extra because they are extra. Okay. Okay, so let's do a quick recap of our notes so we can move on. As I said, we are just focusing on this nucleic acid, which is the DNA, deoxyribonucleic acid. Just what I know about it, firstly, is that it's made up of building blocks called nucleotides. Okay. It's got nitrogenous bases. Now, babe, remember that one? It's got nitrogenous bases, adenine, thymine, which work together and are always the same amount. Cytosine and guanine, same thing. So remember that AT and CG. And then most importantly, your DNA carries the genetic code for protein synthesis, right? Then we look at the brief history, whereby we've got the first two scientists who worked on it. That's Rosalind Franklin and Maurice Wilkins, who researched the structure of DNA in 1952. And then yet came along James Watson and Francis Crick to steal the work. I'm joking. Who actually just built on, but they stole, but they built on the work of these two, and then they used it to now propose a 3D double helix model for DNA in 1953. Which actually reminds me of one of the Big Bang Theory's episode. I think almost the last one, when Amy and Sheldon were working on asymmetry. And then these two scientists now came and were like, well, we proved it, I mean, people will steal your work, so you better be fast about it. Right. So let's continue. Sorry for driving you off. And then now, when the work was then established and people accepted it, Watson and Crick then received a Nobel Prize in 1952, and Franklin had passed on. Right. Then now we look at the shape of DNA, the one they proposed. We just get that, because DNA, DD, therefore it will be double-stranded, two strands. And these strands will be held together by weak hydrogen bonds so that they can be able to unwind. And then because it's in a twisted model or a twisted structure, we call it a double helix. And this is it. Okay, nice one. Which I've also seen in Sheldon's apartment. Okay, guys, I'm a fan. I know. Right. And then this one is just a double strand structure that has already unwinded. And then we can clearly see all of it. Remember phosphate, remember deoxyribose sugar, and the nitrogenous bases. With lastly, the location, DNA that is found in the nucleus is called nuclear DNA, and then DNA found outside the nucleus is there's two places. It could either be in the mitochondria, therefore mitochondrial DNA, or in the chloroplast, therefore chloroplastic DNA, and they are extra. Okay. Also, another place where you can find the location of DNA is my logo. So hopefully you have enjoyed yet again our episode of Just Studying. I appreciate you guys. Again, please continue to liking, to subscribing, to sharing to friends and family, and staying on with us. Thank you.